Hello everyone, welcome to the next video on neoplasia. Here we are going to continue with the previous session. Insensitivity to growth inhibition or the role of tumor suppressor genes. See, normally the cell cycle has some checkpoints, and these checkpoints are controlled by some group of genes called tumor suppressor gene. The most important of them are retinoblastoma and TP53, tumor P53. Fine, yes. So now let us see what is Knudson's two-hit hypothesis. Retinoblastoma is a tumor suppressor gene that is located in the chromosome 13q14. You should remember, very, very important, 13q14, you should remember. Okay, these are the two mutations, okay, which involves both alleles of retinoblastoma, 13q14. Fine, okay, yes. And number two, so what is this Knudson's two-hit hypothesis? Two hits means two mutations which involves both the alleles are required to produce retinoblastoma. This retinoblastoma may be either familial or it may be sporadic. Familial, the child will inherit one copy of the mutated retinoblastoma, right? Okay, and the other copy will be normal. So when this normal copy is mutated only, it will cause this, that is as a result of some spontaneous somatic mutation. Remember children, in familial cases we have two mutation. One is inherited, we call it as familial or germline mutation, and number two is spontaneous somatic mutation. So when the baby is born, he will have only one germline mutated allele. At the same time, when the baby grows, or what will happen, or this retinoblastoma will develop only when the normal allele is also mutated. This is what we call, and the disease is usually inherited as an autosomal dominant trait. This is what we call it as what Knudson's two-hit hypothesis. Remember, in sporadic cases, imagine what will happen. What will happen when the baby is born? Both, there is no germline mutation, only. So if the retinoblastoma has to occur, both the somatic alleles, both the alleles has to undergo somatic mutation, which is not that much possible. That is the reason retinoblastoma is extremely rare in the general population. This is called as Knudson's two-hit hypothesis. Now let us see a very, very important table from this every year one question definite in PG entrance exam. What are the tumor suppressor gene and associated cancer? APC, adenomatosis polyposis coli, okay, protein, okay, it is an inhibitor, okay. So tumor suppressor means it should suppress something. So APC is suppressing WNT signaling, okay, and this is most commonly in colonic cancer, fine, yes. And the second is what we call as neurofibromin 1. Okay. Fine. Then NF1, neurofibromin 1, okay, inhibitor of RAS signaling pathway. It is neurofibromatosis type 1. NF2 is Merlin, okay. This is going to inhibit hippo or pathway signaling. Very, very important. This has been asked in INICET, okay, 2 years back. Hippo signaling pathway or cytoskeletal stability. This is responsible for causing neurofibromatosis type 2. That is schwannoma, meningioma, acoustic neuroma. Then PTCH, patched, okay, it is an inhibitor of hedgehog signaling pathway which is involved in basal cell carcinoma syndrome called as Gorlin syndrome. P10, phosphatase angiotensin homologue, inhibitor of PI3K AKT signaling pathway, P10 mutation, okay, P10 mutation, it is present in Cowden syndrome, P10 mutation. So P10, it is located in chromosome number 10, very easy to remember, chromosome number 10. Then SMAD2, SMAD4, it is an inhibitor of transforming growth factor pathway signaling. This is most commonly caused for juvenile polyposis, and remember this SMAD2, SMAD4 has higher association, higher risk of cancer, pancreatic adenocarcinoma, fine, yes. Then let us see the inhibitors of cell cycle progression. We have retinoblastoma, okay, which is inhibiting G1S transition. This has been asked. Retinoblastoma will act at which phase? It is going to inhibit the G1S transition. Okay, this is responsible for familial retinoblastoma syndrome. Remember, whenever there is retinoblastoma, the most common other malignancy which is present along with retinoblastoma is called as osteosarcoma. Then we have CDKN2A. CDKN2A, it is mainly present in case of familial melanoma. It is a negative regulator of cyclin-dependent kinase. Then von Hippel-Lindau protein, okay, it is an inhibitor of hypoxia-inducible factor HIF1 alpha. Remember, we have a syndrome called von Hippel-Lindau syndrome, which is having a triad of cerebellar hemangioblastoma, retinal angioma, and the renal cell carcinoma. Fine. Yes, STK11. Whenever you hear the word STK11, remember it is Peutz-Jeghers syndrome because it activates AMPK family of kinases. Okay. Yes. Next one is CDH1, cadherin, cell adhesion, familial gastric cancer. TP53. Remember, it will cause cell cycle arrest. Lee-Frommeni syndrome is the syndrome in which there will be TP53 mutation. There will be breast cancer. There will be leukemia. There will be sarcoma. Fine. Yes. Then BRCA1, BRCA2, already I have told you, this is in breast cancer. Then MSH2, MLH1, MSH6, all this is HNPCC, hereditary non-polyposis colon carcinoma. And remember, this MSH2, MLH1, MSH6, all these are involved in DNA mismatch repair. Then WT1 is Wilms tumor 1, okay, that is actually causing Wilms tumor, okay, that is one of the small round blue cell tumors of the infants, okay, or the newborn. And men 1 causes menin, okay, multiple endocrine neoplasia 1, pituitary, parathyroid, and pancreatic endocrine tumors. Okay, yes. So once this is done, this is the pathogenesis of retinoblastoma. I have already told you this is the nuts to hit hypothesis. Remember 13q14. So because of these reasons, the retinoblastoma is called as the governor of proliferation. Okay, whereas T53 is called as the guardian of the genome. Why it is called as the governor of proliferation? Because it is going to act as a checkpoint at the G1S phase. Okay, yes. Okay, I told you when and all the retinoblastoma will happen. So this is see, this is a very important. Just go through this flowchart. What is the key point? What I have to tell you is when the retinoblastoma gene is hypophosphorylated RBC will cause transcriptional block. That means when the retinoblastoma is hypophosphorylated, there won't be any cell cycle progression. At the same time, because of the growth factor stimulus, the retinoblastoma gene will undergo hyperphosphorylation, and that is responsible for transcriptional activation. I remember 2 years back there is a question which has been asked in a PG entrance exam: which of the following is responsible for what the occurrence of retinoblastoma? In that, one of the option was like hypophosphorylated RB, hyperphosphorylated RB. What is the answer? Is hyperphosphorylated retinoblastoma is, or normally the retinoblastoma is not occurring because of which of the following? Then in such a case the answer is hypophosphorylated RB, because hypophosphorylated RB won't cause a cell cycle progression. Fine. Yes. Then coming into TP53 or guardian of the genome, I told you, Lee-Frommeni syndrome, there will be cancer of sarcoma, breast cancer, leukemia, brain tumors, and carcinomas of the adrenal cortex. You have to remember this point. Fine. And we have something called as HPV, human papilloma virus. Human papilloma virus have two proteins children. One is E6, another one is E7. Out of the E6 protein, it is this E6 protein actually binds with the TP53. E6 protein binds with the TP53. Okay, TP53. Okay. Then what are the mechanisms by which this P53 act as a guardian of genome? The number one is it causes DNA damage and hypoxia. Then also it causes oncogenic stress. When these problems won't happen, when this mechanism does not happen, the cell cycle undergo progression. Okay, the cell cycle undergo progression. Fine. Fine. Yes. Okay. Then coming into okay, it will also cause transient P53 induced cell cycle arrest. Okay, and yes, so this is what you are going to explain. So this is how the mechanism. See, a normal P53, whenever there is an oncogenic stress and hypoxia, this P53 will accumulate and binds to DNA. So it will causes transcription dependent and independent effects on the target. It may cause cell senescence, cell death. It may causes P21 and CDK inhibition, which causes cell cycle G1 arrest. Okay, and it will also helps in successful repair and GAD45 and DNA repair. If the repair fails, it will go for apoptosis. Or but at the same time, whenever there is a mutation, what will happen? All these mechanism won't happen. So the mutant cells will undergo expansion. Okay, and they form clonal cells. That is why we call them as a tumor. Okay, yes. Please go through this flowchart. Yes, then this P53 is direct the transcription of several pro-apoptotic genes such as BAX and PUMA. Okay, BAX and PUMA, very very important. They are pro-apoptotic genes. So whenever it promotes apoptosis. Apoptosis means what? Unnecessary cells should die. But whenever there is a mutation of the P53, unnecessary cells can't die. Unnecessary cells can't die. Okay, fine. Then, yes, here comes another important thing. P63 and what is P73? You all will study only P53. P63 is essential for differentiation of stratified squamous epithelium. Okay, stratified squamous epithelium. And P73, it has strong pro-apoptotic effects after DNA damage which is induced by chemotherapeutic agents. Once it has been asked, a question has been asked in JIPMER entrance exam, which of the following genes has a role, or which of the following helps in what the promote apoptosis of the DNA or the damaged cells as a result of chemotherapy? Answer is P73, not P53. Okay, remember it is P73. P63 for epithelial cell differentiation. Okay, yes. Then coming into other things, one is APC, that is adenomatosis polyposis coli. It is called as the gatekeeper of colonic neoplasia. This can be also asked as a short note question. Okay, yes. Remember, APC, it is located in chromosome number 5q21. This also you should remember the band region, everything. It is 5q21. It is 5q21. Fine, yes. And I told you this APC is going to inhibit what signaling? WNT signaling pathway. See, this WNT, it what passes signals through a family of receptors called as frizzled. What is the name of the receptors called as frizzled? That is frizzled. Okay, yes. And this frizzled, we have two protein complexes that is called as beta catenin. See, this is how a normal resting cell will be. When the WNT receptors are there, okay, this beta catenin is going to destruct. Okay, beta catenin has a destructing role. Okay, but so this what will happen? Okay, I will just explain it again. Okay, fine. So WNT signals through a family of cell surface receptors we call it as frizzled, and it will stimulate several pathways. The central one involving beta catenin and APC. So what is the important function of this APC protein is to hold the beta catenin in check. So the APC is going to control the activities of what? Beta catenin. So in the absence of the WNT signaling, what will happen? This APC will degrade the beta catenin and prevent its accumulation in the cytoplasm. So normally when there is no WNT signaling, this APC will degrade the beta catenin. It will prevent its accumulation. So the APC, how it is going to do that? By forming a destruction complex. And the beta catenin TCF, which is another DNA binding factor, which will promote the growth of colonic epithelial cells. So whenever there is the loss of this APC, what will happen? The cell will be continuously stimulated by WNT. I told you, when the WNT signaling pathway is stimulated, APC, if it is normal, it will control the activity of what? Beta catenin. Beta catenin does not undergo accumulation within the cell. When the APC is not there, at that time also what will happen? The WNT signaling pathway will be there, beta catenin will be produced, it will undergo accumulation, but there is no APC because it is mutated. So that is why in beta catenin, what a protein, it's a proto-oncoprotein actually. Fine, yes, this actually undergo accumulation, and finally this will leads to what? Excessive proliferation of the colonic epithelial cells that is responsible for colonic cancer. Then we have E-cadherin. Okay, we have E-cadherin. E-cadherin mainly that is involved in breast cancer. Just remember that. And we have two genes. Okay, one is what we call the snail. Okay, I will tell you. I will explain. This is metastasis also. Okay, what is that? Snail, and another one is what we call as twist. These are the two genes which are involved in which promotes metastasis. Okay, yes. Then CDKN2A, I have already told you. TGF beta is mainly involved in pancreatic cancer. Then P10 is Cowden syndrome, which is an autosomal dominant syndrome located in chromosome number 10. I told P10 means chromosome number 10. Okay, there is a lot of epithelial cancer, endometrial cancer, breast cancer, and thyroid. Then neurofibromin 1, I told you know, that is RAS signaling. The neurofibromin 2 is the Merlin, that is going to cause, I told you, the schwannoma, acoustic neuroma. WAT1, this is located on chromosome number 11. That is very, very important. This is causes pediatric kidney cancer or nephroblastoma. Then patched PTCH is actually it down regulate. Okay, it is inhibiting the hedgehog signaling pathway. Whenever there is a defect in PTCH, what will happen? There will be increased. Okay, yes. Activation of this hedgehog signaling pathway that is responsible for nevoid basal cell carcinoma syndrome, that is also called as Gorlin syndrome. Okay, Gorlin syndrome, PTCH1. Fine. Then VHL, I told you, that is responsible for. VHL is actually going to act as what to say, it is going to inhibit what we call as hypoxia inducible transcription factor alpha, HIF1 alpha. Okay, so VHL, and remember the chromosome number, it is chromosome number 3P. Okay, you should remember this chromosome number 3P. Fine. Then next one is STK11. I told you STK11 means serine threonine kinase. Okay, whenever there is a loss of function mutation of STK11, there will be increased polyps of the GI tract. That too, what we call as Peutz-Jeghers syndrome. You will see in GI pathology. Okay, benign polyps, very, very important. Okay, so I hope this session has been useful. So thank you. We will continue the next in the next part.